So introduction, a fundamental property of living organism is the ability to reproduce. Organisms produce offspring that are similar to themselves. The process occurs by which a single parent cell divides to produce two identical daughters. In this process, the genetic material must be duplicated prior to cell division so that daughter cells receive a full complement of genetic information. The next slide. तो डीएनए रेप्लिकेशन में कौन से एंजाइम्स इन्वॉल्व होते हैं? So this is a list of enzymes involved in DNA replication, but today we're going to discuss about DNA helicase, DNA polymerase, and DNA ligase. The next slide. So DNA helicase. Helicase were discovered in E. coli in 1973 and are a class of enzymes vital to all living organisms. Also known as helix, destabilizing enzyme, they separate the two strands of DNA at the replication spoke behind the topo. They are motor proteins that move directionally along a nucleic acid phosphodiester backbone, separating two annelid nucleic acid strands, that is DNA, RNA, or RNA DNA hybrid, using energy derived from ATP hydrolysis. They have molecular weight of which contains 6 identical subunits. ये अपना हेलिकेस का डायग्राम है. The next slide. Next is DNA polymerase. DNA polymerase are enzymes that synthesize DNA molecules from deoxyribonucleotides, the building blocks of DNA. These enzymes are essential to DNA replication and usually work in pairs to create two identical DNA strands from a single original DNA molecule. During this process, the DNA polymerase reads the existing DNA strands to create two new strands that match the existing one. Also performs proofreading and error correction. So this is how DNA polymerase works. It works as a ऊपर वाला slide. सर slide ऊपर करो. Then the next is DNA ligase. DNA ligase is a specific type of enzyme, a ligase that facilitates the joining of DNA strands together by catalyzing the formation of phosphodiester bonds. DNA ligase is used in both DNA repair and DNA replication. The mechanism of DNA ligase is to form phosphodiester bonds between the hydroxy end of one nucleotide end of another donor. ATP is required for ligase reaction, which proceeds in 3 steps. The next slide. Sir, hello. That was my topic for presentation. Thank you. सर आपका आवाज नहीं आ रहा है.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir. There are 5 DNA polymerase in E. coli. Sir, थोड़ा network issue है. सर नेटवर्क इशू हो रहा है इसलिए आपका आवाज क्लियर नहीं आ रहा. ओके.
Yes, sir. Yes, sir. Sir, is it visible? Good evening, one and all. Myself, Devrao Sharad Sapkar. My roll number is 227325. And today I'm talking about three models of DNA replication. So first we will see what replication is. So in molecular biology, DNA replication is a biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms, acting as the most essential part for biological inheritance. So there were three models for how organism might replicate their DNA, that is semi-conservative, conservative, and dispersive model of DNA replication. So in 1953, a DNA replication proposed by James Watson and Francis Crick. So after that, different scientists give their different opinion on how DNA might get replicated. So some group of scientists says that it follows semi-conservative model of DNA replication. So we'll see the semi-conservative model of DNA replication. It was originally documented through the classical density labeling experiment of Matthew Michelson and Franklin Stahl. Semi-conservative model is that DNA stands separate and a complementary strand is synthesized. for each, so that sibling chromates have one old and one new strand. So as you can see in this diagram, original parental DNA while getting replicated in a first generation, one strand of it have uh, parental DNA and one strand of it has totally newly synthesized DNA. And while getting replicated in second generation, uh, one strand of it has parental DNA and a newly synthesized DNA, and other part of it has totally newly synthesized DNA strand. So the and semi-conservative conservative means only half is getting conserved. Now we will see the conservative model of DNA replication. So David Bloch at Columbia University in New York City, New York, suggested a conservative DNA replication, a theory that involved in manipulation of the DNA bases to avoid unwinding the two strands. In this model, DNA replication result in one molecule that consists of both original DNA strand, which is identical to the original DNA molecule, and the other molecule that consists of two new strands with exactly same sequence of the original molecule. So as you can see, parental DNA while getting replicated in first generation, one strand is totally identical to the parental, and second strand is totally newly synthesized strand. According to the conservative replication model, the entire original DNA double helix serve as a template for a new double helix, such that each round of cell division produces one daughter cell with a complementary new DNA double helix and another daughter cell with a complementary intact old or original DNA double helix. And conservating means it is conserving the property of original parental DNA. Now you will see the dispersive model of DNA replication. So in dispersive model, DNA replication result in two DNA molecules that are mixtures or hybrids of parental and daughter DNA. In this model, each individual strand is a patchwork of how original and new DNA. In 1958, Matthew Meselson and Franklin Stahl proposed this type of replication. As you can see, parental DNA while getting replicated in first generation, some part of it has a parental portion and some part of it has totally newly synthesized strand. So it's like a combination of parental and newly synthesized DNA strand. The experiment done by Messelson The experiment done by the Michelson and Stahl demonstrated that the DNA replicated through semi-conservative model of DNA replication, meaning that each strand in a DNA molecule serve as a template for synthesis of a new complementary strand. The heavier strand represent the parental strand, and the lighter strand is a new one synthesized from a culture indicating the semi-conservative mode of DNA replication. This proves the semi-conservative mode of DNA replication is identical for DNA replication. So we'll take summary now of three models. So in semi-conservative mode of DNA replication, parental DNA while getting replicated, one strand of it has a parental strand and one strand is of totally newly synthesized strand. In conservative mode of DNA replication, parental DNA while getting replicated, one strand is of a fully newly synthesized strand, and one strand is of very identical to the parental one. And in dispersive, parental strand while getting replicated, some part of it has a parental portion and some part of it has totally newly synthesized strands. Thank you, sir. Yes. A heavy isotope of nitrogen K15 and K14. Uh, mi- zinc. Sorry. Oh, so it was in organic form. So first generation में one totally newly synthesized strand and one of parental DNA. Thank you, sir.
Yes, sir. Sir, it is visible. सर नेटवर्क की वजह से गुड इवनिंग एवरीवन माय नेम इज निशा सराटे रोल नंबर 227326 माय टॉपिक इज मिजेल्सन एंड स्टार्स एक्सपेरिमेंट मिजेल्सन एंड स्टार्स एक्सपेरिमेंट Matt Michelson and Franklin Starks originally met in the summer of 1954. The year after Watson and Crick published their paper on structure of DNA. Also, the two researchers had different research interests. They become intrigued by the question of DNA replication and decide to team up and take a crack at determining the replication mechanism. This is three model of the DNA replication: the conservative model, and dispersive model. Introduction. Michelson and Starks conduct their famous experiment on DNA replication using E. coli bacteria as a model system. They, they began by growing E. coli in medium or nutrient broth containing a heavy isotope of nitrogen N15. When grown on medium containing heavy N15, the bacteria took up the nitrogen and used it to synthesize new biological molecule, include DNA. After many generations growing in the N15 medium, the nitrogenous base of the bacteria's DNA were all labeled with heavy N15. Then the bacteria were switched to medium containing a light N15 isotope and allowed to grow the several generations. DNA made after the switch would be made up of N14 as this would have been the only nitrogen available for DNA synthesis. Michelson and Starks knew how often E. coli cells divided, so they were able to collect small samples in each generation and extract and purity the DNA. Then they measure the density of the DNA. This method separate molecules such as DNA into bands by spinning them a high speed in presence of another molecule, such as cesium chloride, forms a density gradient from a top to the bottom of the spinning tube. Density gradient centrifugation allow very small difference like thus between N15 and N14 label. This is N15 is a heavy isotope and N14 light isotope. Result of the experiment. When DNA from the first 4 generation of E. coli was analyzed, it produced the pattern bands of shown in the figure. This is a replication process, E. coli culture, DNA in cesium chloride gradient, and DNA compositions. Starting of replication. DNA isolates a single band after centrifugation. This result made a sense because uh, in the test tube, first in test tube, E. coli DNA ring in down to the test tube. This is heavy isotope of N15. Generation 1. DNA isolated after 1 generation also produce a single band when centrifuged. However, this band was higher intermediate in density between the heavy N15 and the light N14 DNA. The intermediate band told Michelson and Stahls that the DNA molecules made in the first round of replication was a hybrid of light and heavy DNA. This result fit the dispersive and semi-conservative models, but not with the conservative model, because a band for the heavy original molecule and a band for the light newly molecule. Generation 2. Information from the second generation. Let Michelson and Stahls determine which of the remaining models, semi-conservative or dispersive, actually correct. When second generation DNA was centrifuged, it produced 2 bands. One was in the same position as the intermediate band from the first generation, while the second was higher, appeared to be labeled only with N14. This result told Michelson and Stahls that DNA was being replicated semi-conservatively. The pattern of 2 distinct bands, one at the position of a hybrid molecule and one at the position of light molecule. It's just what we have except for semi-conservative replication. In contrast, in dispersive replication, all the molecules should have bits of the old and new DNA, making it impossible to get purely light molecules. Generation 3 and 4. In the semi-conservative models, each hybrid DNA molecules from the second generation would be expected to give rise to hybrid molecules and a light molecule in the third generation, while each light DNA molecule would only yield from more light molecules. Thus, over the third and fourth generation, we have accept the hybrid band to become progressively fainter, because it would represent a smaller fraction of the total DNA, and the light band to become progressively stronger, because it would represent a larger fraction. As we can see in the figure, Michelson and Stahls saw just this pattern in their result, confirming as semi-conservative replications models. Conclusion. The experiment done by Michelson and Stahls demonstrate that DNA replicated semi-conservatively, meaning that each strand in DNA molecule serves as templates for synthesis of new complementary strands. Although Michelson and Stahls did their experiments in the bacterium E. coli, we know today that semi-conservative DNA replication is a universal mechanism shared by all organisms on a planet Earth. Some of your cells are replicating DNA semi-conservatively right now. Thank you, sir. In E. coli sample, DNA extract cesium chloride in cesium chloride density centrifugations. This is measure. Generation 2, sir. Generation 2 because gen Be- because dispersive replication, all molecules should have bits of old and new DNA. Thank you, sir.
Yes, sir. Okay, sir, are you able to see? Okay, good evening, everyone. My name is Shreya More, and today I'm going to present on the topic John Kane's experiment. So, who was John Kane? He was a British physician and molecular biologist who made significant contributions to molecular genetics, cancer research, and public health. He was born in the year 1922 and died in 2018. So Keynes discovered new pathways for DNA repair. So this is the Keynes experiment. So the visualization of replicating chromosome was first accomplished by John Keynes in 1963 using the technique called autoradiography. So autoradiography is a method of detecting and localizing radioactive isotopes in macromolecules by exposure to photographic emulsion that is sensitive to low energy radiation. So it is used particularly in studying DNA metabolism because DNA can be specifically labeled by growing cells on thymidine, the titrated deoxyribonucleoside of thymidine. Thymidine is incorporated exclusively in DNA and it is not present in any other major component of the cell. So what was the procedure of the experiment? Basically the cells are grown in a solution containing radioactive thymidine. So then the surface is immersed in a radioactively sensitive emulsion containing silver bromide. Silver bromide is used as it is sensitive to light. So the titrated thymidine is incorporated into the chromosomal DNA of the cell. So then the chromosomes are isolated by gently lysing the cells and fixing the chromosome to a photographic surface. The radiations after that released from the titrated thymidine converts silver ions in silver bromide into insoluble metal green. So after a following period of exposure, excessive silver bromide is washed away, only leaving the silver grains to appear as small black dots. So when the photographic film is developed, it can be visualized with an electron microscope. So this was the procedure. So So what is chromosome length? John Keynes pioneered a technique for measuring the length of DNA molecules by autoradiography. Previously, chromosome length was not able to measure as it was in a condensed form during mitosis. So Kane used autoradiography to visualize the chromosome uh, whilst uncoiled, allowing for more accurate indications of length. By using titrated uracil, regions of active transcriptions can be identified within the uncoiled chromosome. So this was the picture of the uncoiled chromosome identified with autoradiography before transcription and during transcription. So as you can see, during transcription, it has become a little bit dense. So John Keynes was further able to use autoradiography to demonstrate key events which occur during chromosomal replication. So DNA replication involves formation of a replication bubble. So how it is formed? So one strand of DNA is replicated into two identical copies. So in order to occur, DNA must be separated with an enzyme known as helicase, like a zipper that has an unzipped in the middle. The separated DNA strands from a little open buckle. So this was the replication bubble. So what is replication bubble? It is a structure formed by the separation of two DNA strands by the helicase enzyme. So basically in eukaryotes, there are several replication on each chromosome. The two sides of each bubble goes from zipped to unzipped, and it is known as the replication fork. So after the replication bubble is formed, there are some special proteins that are known as Ssb protein, that is single stranded binding proteins, that binds to individual strands and prevents the helicase from binding up again. So in bidirectional replication, the replication process occur in two direction, basically unidirectional and bidirectional replication. So in unidirectional replication, the process occur in one direction, and in bidirectional replication, the process occur in two direction. So in unidirectional replication, only one replication fork is formed, whereas in bidirectional replication, two replication fork is formed. In unidirectional replication, only one end of the replication eye is moving or growing, and in bidirectional replication, both the ends are moving. So these are some references. Thank you. Yes, thank you, sir. As it was, so basically it is an image of an X-ray or a nuclear emulsion produced by the beta particles or gamma rays. Thymidine. It is particularly used to label DNA. H3 thymidine. I don't know, sir. H what? H2. Okay. They are particularly the hydrogen bonds. Thank you.
Yes, sir. Hello, sir. Uh, So, my name is Pratimesh Kavre, and my topic is enzymology of DNA replication. Hello, hello, hello. Huh. So, my topic is enzymology of DNA replication, helicase, DNA polymerase, and ligase. So, DNA is a double-stranded molecule held together by hydrogen bond in order to read and replicate the DNA sequence. An enzyme called helicase must read separate the strands by breaking the hydrogen bond. This creates the gap between the two strands called replication bubble. The point at which the two strands begin to separate is called the replication fork. The enzyme that read the DNA and create a new copy is called the DNA polymerase 3. This enzyme continuously reads DNA in the 3 to 5 direction, traveling away from the replication fork. The DNA strands that runs from the fork in the 3 direction referred to as the leading strand. Because of its directional dependence, DNA polymerase 3 can only read the other DNA strands in short section, traveling in the 3 direction towards the replication fork. This template DNA strand is referred as to the lagging strands. DNA polymerase 3 is reading the 3 to 5 strand and creating the new DNA in the 5 to 3 direction. This means it is adding nucleotide to the 3-hydroxyl end of the new DNA strand being made. The new thymine base is being added to its complementary base pairing with adenine. When DNA polymerase 3 adds a new nucleotide to the DNA strand, it forms a covalent phosphodiester bond between the adjacent nucleotide 3-hydroxyl group and the new nucleotide 5-phosphate group. On the lagging strand, the new DNA strand is being created in the direction away from the replication fork in small section intercepted by short RNA primer laid down by the enzyme primase. The lagging strand can only replicate in small section due to 5 to 3 directional dependence of DNA polymerase 3. These small sections are called Okazaki fragment. This means the replication force need to be repeated several times. Then the DNA polymerase 1 has to go back and replace the RNA primer with DNA. The gap between these small replicated sections needs to be prepared and sealed. This last step is performed by DNA ligase, an enzyme that creates an unbroken phosphatase sugar backbone. Thank you, sir. Hello. Yes, sir. किसके पास है? Hello, sir. Hello. Sir, DNA polymerase 1. मालूम मालूम नहीं सर.
यस सर. सर मैंने आपको भेजा है पीपीटी. Yes, sir. Good evening, everyone. My name is Tanushri, roll number 227329. Today we are going to see about enzymology of DNA replication. So what does enzymology means? It is a branch of biochemistry that deals with study of enzymes based on its function, properties, or significance activity. Next slide. DNA replication, it is a process where a genome's DNA is copied in a cell, means a complete set of genetic information is stored in organism. And replication of DNA is a complex process that involves a number of enzymes to work. There are many steps like temporary separation of two parental strands, stabilization, initiation, elongation, and termination. As we all know that there are many enzymes like topoisomerase, Ssb protein, Ssb protein, DNA helicase, DNA polymerase. Today we are going to see about topoisomerases. Next. Topoisomerases, DNA topoisomerases or topoisomers are enzymes that catalyze change in the topological state of DNA, interconverting relaxed or supercoiled forms, linked, catenated, and unlinked species, and knotted or unknotted DNA. Topological issues in DNA arises due to an intertwined nature of its double helicase structure. For example, this can lead to overwinding of DNA duplex during DNA replication and transcription. As we all know that topological issues arises in DNA due to interwinding of nature of double helix. As sir has given us example that if we take two rubber bands and we go on overwinding it, then it will means pressure will occur on that. The same way, the DNA also will produce. If it left unchanged, this torsion would eventually stop the DNA or RNA polymerase involved in this process from continuing along the DNA helix. A second topological challenge result from the linking or tangling of DNA during replication. Left unresolved link between replicated DNA will impede cell division. Impede means it will delay or delay cell division. And tangling means it will twist together. Next. So these are two types of topoisomerases. Topoisomerase 1 and 2. Topoisomerase 1, it cuts only one strand, and 2, it cuts both the strand. And then topoisomerases maintain several, means it serves to both transcription and replication of DNA. The DNA topoisomerases prevent and correct these types of topological problems. They do this by binding to DNA and cutting the sugar phosphate backbone of either one type, means either of topoisomerase 1 or 2. This transient break allows the DNA to untangle or unwound, and at the end of this process, the DNA backbone is released. Since the overall chemical composition and connectivity of DNA do not change, the DNA substrate and products are chemical isomers differing only in the topology. Next. So now Ssb protein. It is also known as single-stranded binding protein. During DNA replication, the single-stranded DNA binding protein wraps single-stranded DNA with high affinity to protect it from degradation and prevent secondary structure formation. Although SSB binds single-stranded DNA tightly, it can be repositioned along single-stranded DNA to follow the advancement of replication fork. Using all atom molecular dynamics simulation, we categorized the molecular mechanism of single-stranded DNA associated with SSB, placed in solution a single-stranded DNA and SSB assemblies were observed to change their structure spontaneously. Such structural changes were suppressed in the crystallography environment. Means DNA gets suppressed and uh, DNA DNA suppressed and crystallography environment occurs. Next. TUS protein. TUS, also known as terminus utilization substance. It is a protein that binds to terminator sequence and act as the counter helicase. When it comes in contact with an advancing helicase, the bound TUS protein effectively stops or halts DNA polymerase movement. TUS helps and TUS helps end DNA replication in prokaryotes. Thank you. TUS. Hello, sir. Terminus utilization substance. किसके साथ? It binds to a terminator sequence. Catenated DNA means two circular DNA strands are linked together like a chain in chain structure में. Resolved. Means उसका मीन्स प्रेशर अगर अपन ये करेंगे वैसे ना, नहीं पता सर। थैंक यू।
हेलो सर। मैं प्रेजेंट करूं? यस सर। ओके। हेलो। और स्क्रीन दिख रही? ओके। यस सर। So my name is Bhagyeshri. Roll number is 19. My topic is DNA replication in eukaryotes, Saccharomyces cerevisiae. So first, what is DNA replication? Replication of DNA is vital to the transmission of genomes, and the genes they contain from cell generation to cell generation and from organism generation to organism generation. The biochemistry and molecular biology of DNA replication are similar in prokaryotes and eukaryotes, but in eukaryotes is that DNA is distributed among many chromosomes rather than just one. DNA replication is semi-conservative. Each DNA strand serves as a template for the synthesis of new strand, uh, producing two new DNA molecules, each with one new strand and one parental strand. This is semi-conservative replication. And what is replicons? The stretch of DNA from the origin of replication to the uh, two new terminal of replication on each side of the origin is called replicon. It's also called unit of replication. The E. coli genome consists of one replicon of size 4.6 MB with a rate of movement of each replication fork of about 1000 base pair per second. Replicating the entire chromosome takes 42 minutes. Each eukaryote chromosome consists of one linear DNA double helix. Replicating the entire genome takes 8 hours, but each replicon is replicating for only one part of that time. And DNA replication in eukaryotes divide into 3 stages. First is initiation, second elongation, and third termination. Diagram This diagram shows three steps. First, initiation step is happen in G1 phase. Second, elongation is also happen in S phase, and last termination is happen S phase. So, initiation of replication in eukaryotes. Replicators are less well defined in eukaryotes than in prokaryotes. The identification of the ORC, origin recognition complex in Saccharomyces cerevisiae, was an important advance in understanding eukaryotic DNA replication. It was identified by Stephen Bell and Bruce Stillman in 1992 as a factor that's specifically bound to the yeast uh, auto- autonomously replicating sequences. In the yeast, Saccharomyces cerevisiae, replicators are approximately 100 base pair sequences, and it's called ARC, autonomously replicating sequences. The initiator protein is in eukaryotes is the multi-subunit origin recognition complex. The yeast replicator, for example, spans about 100 base pair. The ORC binds to the two different regions at one end of replicator and recruits other replication proteins, among which is the protein needed for DNA unwinding in a third region near the other end. The origin of replication is between the first two regions and the third region. Thank you. Yes, sir. सॉरी सर, उस टाइम मैं तबीयत ठीक नहीं थी. DNA replication speed. Uh, 100,000 base pair, 1000 base pair per second. Mm, 10 raised to 6 something. Mm. तो नहीं पता सर. एक साथ होते हैं. थैंक यू.
Thank you, sir.
Thank you, sir.
Thank you,
Yes,
sir.
Priya.